Welcome to Miller Bio, starring the cell cycle. Today we'll be looking at cell cycle and mitosis. And we've talked about the different types of cells that an organism like yourself has in their body. And we're going to look at the life cycle of those cells. So your cells do you a favor. Because they will die within the time of your life, and you need other cells to replace them, before a cell gets too large or dies, it will divide. And the process of this cell division going into two smaller cells is the process of forming daughter cells. So daughter cells are exact copies of the original parent cell, and they're genetically identical. So before the cell divides, it will make copies of the genetic material. DNA, again, deoxyribonucleic acid, which is a type of macromolecule called a nucleic acid, stores the genetic information for how to build you. DNA that's found in your body is tightly coiled and held together with proteins. And these proteins serve as a way to wrap this DNA into a tightly coiled configuration. It's called the condensed form of DNA. And DNA in its original form, when it's not condensed, is called chromatin. So when the DNA condenses into chromosomes, it consists of two identical structures called the sister chromatids. So in other words, this sister chromatid shows DNA that's tightly wound around itself and around these proteins, and it forms this X-looking shape where we have loads of DNA wrapped in that X-looking shape. So these identical structures are called sister chromatids, each one, and there's a centromere in the middle that holds the two sister chromatids together. So as we look at the life cycle of your cell, we see that the cycle begins in this part here where we've got our cell, and it goes through this big long phase called interphase. And that is where, if we look at all of your cells in your body right now, we'd find that the majority of them are in interphase. Why? Because interphase takes up the longest period of time of that life cycle of that cell. Now, as it approaches cell death, the cell will copy itself into two daughter cells. And that's what we call mitotic phase. So this small slice of the pie is dedicated to what we call mitosis, and then the other small piece at the very end is called cytokinesis, which is where there's actually a pinching off of those new cells. Now interphase is split into three parts: G1, synthesis, which stands for DNA synthesis, and G2, and we'll break each of those down as we go. So what is the cell cycle? A series of events that the cell goes through as it grows and divides. There are four phases, and that's what we looked at here. So we've got G1, synthesis, and G2, and then the mitotic phase. So interphase, which is a period of cell growth, that's what we said lasts the longest, consists of the G1, S, and G2. M phase is the actual nuclear division. So that's this part right here. So again, 1, 2, 3, 4. And this is where we're going to spend a lot of our time, because this is where a lot of the action happens, is in M phase. And M phase consists of mitosis and cytokinesis. So specialized proteins called cyclins help to regulate the cell cycle as well. Again, here's another graphic showing the timeline of events that takes place. We go G1, in which the cell grows, S, in which DNA is copied or replicated, G2, the cell prepares to divide, and then within M phase, we have mitosis and cytokinesis, where the cell actually divides. So gap 1 phase is within interphase, and this is where the cell does a lot of its growing. This is an increase in size, and this is a production of new proteins and organelles. The synthesis phase, which is the part 2 of interphase, is the main process during which the DNA is synthesizing or replicating itself. So DNA is replicated so that the daughter cells will have the exact same amount and type of DNA as the parent cell. If you had a cell that is supposed to be identical to the one that is dying and leaving its legacy, so to speak, but it's not the same genetic makeup, that's a problem for you. So we want to make sure that there is accurate replication or copying of that DNA to go to the new cells. Gap 2 here is the shortest of the 3 parts of interphase, and generally what happens here is that the organelles and molecules needed for cell division are produced. So those parts that are specifically going to be needed in M phase are going to be produced during G2. And then, the cell goes from interphase into M phase. So the phase of cell division is mitotic phase, and M phase, or mitotic phase, consists of 2 parts, mitosis and cytokinesis. So mitosis is the division of the nucleus and the DNA that's contained in there. Cytokinesis is physically the division of the cytoplasm, where there's actually a splitting of 2 cells where they pinch off. So once M phase is finished, the cell cycle is complete and will begin again in the 2 newly formed daughter cells. So the 2 newly formed daughter cells then begin this phase of their own life cycle. So here we go again. Here's the basis for the cell cycle and the way that each of your cells undergoes its life cycle. Interphase consisting of G1, S, G2, then going into M phase, which consists of mitosis and cytokinesis, and yields 2 daughter cells where we started with just 1 parent cell. This is where we're going to look next, which is as we enter into mitosis, what happens in the cell as it prepares to divide. Now, we leave off in interphase here. This is G2, cell is preparing to divide, making up the parts that it needs during division. And then you notice that as G2 comes to a close, the nuclear membrane is beginning to the DNA within is beginning to condense into these chromosomes. And then during prophase, which is the first stage of M phase, so again, we're in M phase within mitosis, and now we're going to break it down into several phases, the first of which is prophase. You'll notice that here the chromosomes consist of the 2 sister chromatids. They're condensing from this chromatin, stringy DNA, when it's in the resting phase. And then the nuclear membrane is going to begin to disintegrate or go away. Why is this happening? Because the DNA is about to be split into 2 sides of the cell, and eventually at the end of all this we're going to end up with 2 new daughter cells that are genetically identical to that original parent. Now in the process we go from prophase to metaphase to anaphase, and then telophase, and lastly cytokinesis, and this is that pinching of the 2 daughter cells into 2 new cells. Metaphase is known for the lining up the chromosomes along the midline, and anaphase is known for the beginning of the separation of these identical sister chromatids. So what's on this side of the cell is going to be genetically identical with what's on this side of the cell, making 2 new daughter cells, each with the same exact number and type of DNA from the original parent cell.